Good morning to my dear students. Students, today we are going to discuss our first chapter of plant physiology, photosynthesis in higher plants. So, students, as we all know that photosynthesis is a very important process which is regulated by enzymes, and it's a very important anabolic process, means constructive process in which the complex organic food is going to be synthesized. And this process is taking place basically in the green parts of the plants, which contains basically a green colored pigment, chlorophyll. And in this process, as we all know that food is synthesized with the help of carbon dioxide and water. And we can say that this is the process, or this is a photosynthesis is a chemical process in which solar energy is stored, and it is converted into the chemical energy in the form of the food. And I want to tell you that this process is very important process because it includes two important steps. The first step is called production of primary food in the form of the carbohydrate or glucose. Second, why it is important? Because it is responsible for the restoration of oxygen gas into the ecosystem. So we can say that photosynthesis is a biochemical process, or it's an anabolic process which involves the formation of or synthesizing of organic food with the help of carbon dioxide and water in the presence of sunlight. And students, today we will discuss what are the various experiments performed by various scientists while the discovery of photosynthesis was going on. So the initial credit goes to Joseph Priestley, and during 1770 he conducted a series of various experiments with the help of a bell jar, candle, and mouse. So what he did, I am telling you. The Priestley observed that a candle burning in a closed bell jar soon gets extinguished. Similarly, while a mouse is kept in a bell jar or closed bell jar, so definitely after some time it will get suffocated, and it will also die. And while the both candle as well as the mouse are kept into inside a bell jar, so somehow we have to say that they are uh, damaging the air, or they are while the animal is breathing, at the same time he is damaging the composition of air, as well as while the candle is burning, at that time it also damages the air. Due to it, uh, the, the main gas which is required for the burning of the candle as well as survival for a living animal is required is going to be damaged out, and that's why the candle is going to extinguish, as well as the mouse is getting suffocated and finally dying. So then what he did in his experiment, Priestley placed a mint plant in the same bell jar when a burning candle was there, or as well as a mouse was there, and he observed that in the presence of the mint plant, the mouse stayed alive as well as the candle continued to burn. And then finally, Priestley hypothesized that the plant are the main organisms which are restoring the air, whatever was disturbed or damaged by the breathing animal as well as the burning candle. Here in the picture you can see that the first image at the first image there is a bell jar, closed bell jar, and inside it a burning candle as well as a mouse is plotted or placed. And after some time you are observing both of them are, mouse is going to be die as well as the candle is going to be extinguished. While a plant is plotted in the third one diagram, you can see that along with the burning candle as well as the mouse. So after a few interval of time we are able to see that both means mouse is alive as well as the candle is continued to burning. Then after the second experiment was done by Jan Ingenhouse, and during his life cycle 1730 to 1799, and with the help of his experiment he showed that sunlight is essential for the plant, uh, Actually, photosynthesis is the process in which the plants are going to purify the air, and Jan Ingenhouse during his experiment proved that uh, sunlight is essential for that particular process. Means the process in which the plants are going to purify the air, in that for that particular process sunlight is essential. And how he performed his experiment, I am telling you that for his experiment he chose aquatic plant, and he showed that the bright sunlight, while the aquatic plant is kept, small bubbles are going to be arise. Okay, while the same plant is kept into the dark room, no bubble formation occurs. Here in the picture you can see that while a aquatic plant is kept inside a funnel and an inverted test tube is there, so here we are observing small bubbles are going to be formed. And while the same experiment is done in the dark room where there is no sunlight, so no bubble formation will occur there. Later on he identified that these bubbles are of oxygen gas. Hence, he showed that it is only the green part of the plant that could release oxygen. So, finally, he concluded that during this process, the bubbles which are coming out are of oxygen gas, and this process takes place only in the green parts of the plant. Means sunlight is essential. Along with the sunlight, green parts, this process only occurs into the green parts. Now the third one experiment was done by Julius von Squash in 1854, and he proved with the help of his experiment he proved that the plant produce glucose while they are growing, and the glucose is finally stored in the form of the starch. And he also explained that uh, the plant carries a green pigmented or green substance, okay, which is present in specialized cells where this glucose formation occurs. Okay, and then after T.W. Engelmann uh, performed one more experiment. Actually, what he did, he splitted the sunlight with the help of a prism. As we know that the sunlight is composed of 7 different colors, Vibgyor, and he used a green algae Cladophora and placed it in the suspension of aerobic bacteria. Actually, what are aerobic bacteria? The bacteria which are going for the aerobic respiration, means they will require oxygen, and a green algae Cladophora will perform photosynthesis and they will form oxygen, and with the help of this oxygen the bacteria can go for the aerobic respiration. And Engelmann spotted that the bacteria are especially accumulating at the uh, blue as well as the red spectrum of the light. So it means the chlorophyll A and the chlorophyll P pigments, okay, which are providing green color to the plant, okay, and these pigments are responsible for absorption of solar energy and conversion of solar energy into chemical energy. Or simply I have to say that these are the pigments which are mainly functioning for the synthesis of the food. And these two pigments, chlorophyll A and chlorophyll B, are most active in the uh, spectrum of the blue light as well as the red light. So he concluded that out of the 7 colors of the sunlight, blue color and the red color of spectrum is most essential for the photosynthesis. Okay, and the key features of the photosynthesis was first explained by the 19th century, and the main features were that in case of the plants, the light energy is utilized during the photosynthesis, and during the photosynthesis carbohydrates are synthesized from carbon dioxide and water. And empirical equation was given, and this equation is CO2 plus H2O. You are observing one arrow, and in the top of the arrow light is given, so it means the presence of light is essential. And here you are observing CS2O is mentioned, and CS2O means it is representing basically carbohydrates, and oxygen gas is going to be formed as a product. So here initially the equation for photosynthesis was given, and this equation is CO2 plus H2O. In the presence of sunlight they are combining together and they are forming oxygen gas as well as the carbohydrate. But later on Cornelius von Neil did one more experiment with the purple and the green bacteria, and he demonstrated or he concluded that photosynthesis is a life dependent process. And he explained that during this reaction uh, carbon dioxide is going to be reduced. While reduction is there, the definitely reduction means addition of hydrogen, and this hydrogen comes out from the splitting of the H2O, means water is going to split out and the hydrogen releases, and this hydrogen which is released from the splitting of water molecule, it reduces carbon dioxide and forms carbohydrate, and the oxygen content in the form of the oxygen gas is released into the atmosphere. So finally he concluded that the oxygen which is going to be released into the atmosphere during the photosynthesis comes out from the water, not from the carbon dioxide. And the correct equation for the photosynthesis was given: 6 CO2 plus 12 H2O. C6H12O6 means glucose or carbohydrate, plus 6H2O plus 6O2. So this is the final equation for the photosynthesis, and these were the different experiments for the uh, in the field of the discovery of photosynthesis. So I am concluding myself today. I hope so you understood what is photosynthesis and what are the various experiments performed by various scientists. Okay, once again I will meet you in the next video. Till then, thank you, bye bye, and have a good day.